discussion just with a kind of, well, asking the panelists to do a quick, I mean, you already met some of them, but like a quick small introduction of themselves, but I think also to highlight an anecdotal kind of example of a success other than kind of maybe the ones that people all know about of AI in helping kind of healthcare from a genomics setting.
Oh yeah, this is a great pleasure to start this off. So my name is Fritz Hirschek. I'm associate professor at Baylor College of Medicine. Great success of AI. There has been a few. I mean, like, we are using it a lot for base calling and long reads and single nucleotide variant calling long reads. These models are really getting refined and improved and improved. I think there's still a lot of development in there. I think it was kind of exciting to see AlphaFold and these kind of developments. But like, I don't think many people have bridged the gap between like genomic variants and then really protein consequences. Also because maybe or maybe not AlphaFold is a little bit overhyped because of the whole thing. So yeah, I think that's kind of it.
Hi everyone, I'm Stephanie Hicks. I'm an associate professor at Hopkins in biostatistics and biomedical engineering. My work is in genomics. You'll hear all about that later. I think for me, a success that I have seen AI being used outside of pharmacogenomics is in my own area of research, using deep learning models to be able to do unsupervised analyses. So a lot of AI has been developed in the world of supervised analyses, and there's a big push in the field for what's called self-supervised or semi-supervised analyses. Much of genomics is often entirely unsupervised. Like, we go into a new region of the brain, we want to discover what the patterns are in the brain, we want to figure out what the genes are that are like driving the variation here. So all my work is almost always unsupervised. And I've been delighted to see AI, and specifically the use of self-supervised or semi-supervised methods, have such a great launch in these areas. And I can talk about that if anybody wants to know more.
So I am Sonia Villapol. I'm assistant professor in the Department of Neurosurgery and the Center for Regeneration at Houston Methodist Research Institute. And yeah, so I basically, in my lab, we have clinical studies. So that means we work with doctors, with physicians on this side, looking for biomarkers for any neurological disease, but basically Alzheimer, long COVID, traumatic brain injury, concussions, and all that. So I'm kind of jumping between our collaborators, my informatics side, and as well the physicians, and that gives me some quite perspective what they want, or where are the needs and what we have to improve, and we can discuss that later. And yeah, I think that.
Blake Hansen, I'm an assistant professor at UT in the School of Public Health with a joint appointment in pediatric infectious diseases. And I think probably the coolest observation that I've seen using, and I'm going to differentiate true AI from more of a machine learning and kind of refinement type method, but being able to utilize genomics and surveillance across multiple hospitals and being able to identify cryptic transmission networks that point to things like biofilms in the sink drains being disturbed in certain situations and causing infections, I think are really, really fascinating ways to try to identify and then be able to act upon these different pathogens and environments and be able to really help people stop getting infections in the hospital. It's a place you don't want to be unless you really have to be there. So everything we can do but from there, and I think there's some really cool machine learning based methods that are coming out that are refining exactly how to do that.
Okay, so I think that actually sets the stage really well for, I think, our first question. So we talked about successes, but then the question is, in your guys' opinion, what are the pain points that keep genomics analyses from playing a larger role in healthcare today? So, whoever.
I can start that.
Okay.
There we go. So we do a bunch of this, and that's always the goal. So we have a number of different tools. We have one I'll plug called GenoStellar, which is actually a molecular biotyping and kind of analysis platform for Klebsiella pneumonia that we're expanding out. But there's kind of three major pain points that we have really identified. One is analytical capacity. So this is still very tough to analyze this data. It requires a lot of expertise. I mean, I just showed you all the things we're still dealing with and kind of trying to refine that makes this difficult to implement. I just don't think we quite have trained the generation of people that will apply these things in a clinical environment like you would see for like a clinical micro lab yet. Another one is cost per sample is still pretty high, even with the cheapest methods that we have. If you want to sequence every bacterial infection isolate in a clinical environment, they're going to laugh at you because it is so cost prohibitive. And I think the third one, and this is where we get into how do we apply this more for things like treatment and diagnostics, is that genotype-phenotype correlation is still not totally solved. So that example I showed of Will Shropshire's data with that amplification of that transposable unit, that is something that, unless we had specifically looked at that, we would not have understood why that organism was phenotypically resistant. And if you were to run a genotype screen and say, try to predict resistance based on the information without taking into account copy number variation, you're going to miss. And so we still have a lot of work to do to really refine how do we use genomics, potentially in combination with proteomics or something else as well, to be able to refine and say replace culture-based susceptibility testing. A lot of people are working on it, but we're not there yet because of that genotype-phenotype gap.
Oh, excellent. It's like I don't have anything. No, but yeah, something that you didn't mention for sure that you will be agree about is the trash. Like you know, there are a lot of cofactors and all that can influence and the genomic data and everything. And so the trash from the physician side, that this is valid or what kind of other factors they can impact and make an influence. So for us is the more challenging maybe, and also as well, of course. And the grants reviewers and papers is kind of translated to the clinicians, to the reviewers. But I think that this is some challenge that we have to work on that, the way that we can show and interpret the data for everyone, you know, for the public, but especially for the physicians, so they can believe that the thing that we're doing makes sense.
I would echo that wholeheartedly. For me, in my own world of single cell and spatial genomics, I would say much of the field is still very focused on generating an atlas or a map. And that's great. I mean, that's like kind of the first step, but that's very far from the clinic or any kind of like translational aspect. I think basic researchers have kind of like the opportunity to work very hard on generating these atlases and then kind of like say, here we go, this is what I've discovered, and then they kind of leave it there. And then there's like this huge gap for now, what do you do to take these atlases, or how do you move beyond just like gene signatures to be able to get it to that preclinical stage or that clinical stage. So I think the field itself, that's just kind of where it is. It's still in the state of how do we generate these big atlases, which again, very important, very exciting, but there's still a long way to go past that point. So, yeah.
Yeah, for the sake of fun of it, I disagree with you guys. So one point that you mentioned was cost. I don't think that this is actually a big factor. So like in the study that we did, I think our average costs were like $5,000 to $6,000 per patient, but that was on this rapid segment. This is nothing compared to an MR scan or something like that, right? So I think, in my opinion, what's prohibitive is like, show me that it works, kind of, right? Because, I mean, like, clinical sequencing right now, and I'm just thinking about human genomics, right? Not metagenomics, which is, you have a good point of like, that's probably a different mindset. We have a diagnostic rate of 15-20%. This is chicken shit. And then you have other metabolistic measurements that work much better. I mean, like, if a guy has a fever, then give him something against the fever. That's how medicine works these days. We don't need to sequence that guy. But like, once that person, and I'm not targeting you per se, I'm just saying, like, just for the sake of argument, to have a little bit of fun here. And like, once everything else failed, right, then they come to like, oh, let's try sequencing, and this is going to be a miracle machine. But obviously it's not, and lots of it is also what you guys said is, of course, this genotype, phenotype. Or I would just generalize it like lots of the mutations that we are seeing right now, especially in adult diseases, we don't have good control groups to kind of understand these mutations. So there's still a lot of like building a reference up or a reference setup to better understand these variants. But like, there are also some very promising things, and I think sequencing, especially on the clinical side with exon sequencing or panel sequencing, is getting more and more adopted because like the cost is not that much of a problem. There's still some discussion on how to kind of refinance over the medical insurer, but that's more bureaucratic hurdles. So I really think it's more of the point of like showing that it really works better or the same level as MR scan or something like that.
Yeah, I guess I'll clarify one thing, 'cause I think you're spot on, is that the human genomics part of it, and particularly cancer genomics, you can reimburse the insurance companies for it. Bacterial, I don't know of a single example where I can get them to pay for anything. So it's more like even at $50 an isolate, you do 30,000 of those over a year, it becomes a very, very large number. And so that's more like we haven't proved that it works well enough to get anybody to agree to pay for it, I think is the big thing.
But on your universe, like, could you showcase this over secondary infection reduction, or like you think more about like infectious disease in general when the patient comes in with it, or like?
You've hit exactly on what my next grant application is, which is demonstrating this from like an infection control point of view as a way to get in the door and then build the models, be able to reinforce the data sets, and then pan out from there. Because if someone gets an infection and they all of a sudden can't charge for the $400,000 hospital stay, they're going to get really happy if you can prevent infection.
And then what always fascinates me a lot is like to look at it from the other perspective, right? So I remember a case in my PhD time. There was a doctor in an Austrian hospital, nevertheless. And so he was happy with looking at his, 20 genes, right? And so we proposed to do a whole genome or like add another 20 genes, and he was absolutely against it. And it was not, he saw the potential benefit of like improving the medication or improving the treatment, but that was not his problem. The problem was actually that he was scared the hell out of that we now find new mutations in these genes that he hasn't been able to report before to the patients, and then get sued over it because like he didn't found this mutation. No, it's a real thing, but people don't discuss these things in that setting. So like I think there's a lot of scareness as well in like medical providers of opening up to these regions that we haven't touched so far. But like if we do whole genome clinical, like we would no matter what touch this, and then the whole thing goes from like what do you report out? That's a whole another discussion, of course. But like there are some other interesting driver things that really play into this question of adopting the sequencing thing.
And as a really cool example of that, just briefly, 23andMe ran into this where they were actually able to detect people who had cancer in their blood based on panels and number of chromosomal copies. And so there was a whole discussion of, okay, we didn't design the panel to do this, but if this person knows they can get treatment, are we able to report, and then ethically, do we have to report because they can find these things?
Yeah, we have the same discussion right now. So with that somatic mosaicism grant that we just have, which is kind of 150 individuals, going to be sequenced like 7-8 tissues. Obviously, these people are dead already from a non-natural cause, most likely. So we are sequencing their brains, we are sequencing their guts, we are sequencing their hearts, whatever, right? So what do you do if you find cancer mutations, right? And this was like hours of discussions just for you guys to appreciate it, because like, well, now we know that this guy had pre-cancer mutations, right? So do we go back to its spouse and tell her that there were pre-cancer mutations in an individual that she's, well, related but not blood related, right? So make her life miserable. But what happens if they have kids, right? The kids probably want to know. So like, how do you handle this then? Holy shit, right? There goes the Christmas. So like, I just think like often it's easy to say like costs or like reporting or whatever, but like there are really, really deep discussion points that have to be solved in order to really kind of apply it on a very broad scale.
Yeah, so it's actually really interesting because I think there was actually a question about cost of sequencing, and I guess you guys actually inadvertently answered that question also in this discussion. But actually, I think the next question is actually also kind of related, and thinking about like having people kind of trust results, or think of it like an MRI. So the question is, what are your thoughts on improving reproducibility in genomics? It seems like integrating more ML and AI may make transparency more difficult.
It's a mess. I don't know. It's very so. I mean, clinical sequencing, as constrained as it is, it has some certain form of reproducibility. It's not like, I think there was a really nice Galaxy paper from, I always forget his name, from James Taylor, yeah, and his group, sadly he passed, that showed like what, 2% reproducibility in sequencing studies, at least in research settings, right? So that's a nightmare. And that was a big motivation how Galaxy came around. And it is a big issue. There was another side question, how to trust the results or something
Well,
like
I
that?
think it's just like this general comment. Sorry, I
Sorry.
said that the question was answered already. So it says, integrating more ML and AI may make transparency more difficult. So what are your thoughts on improving reproducibility in this kind of context that we're...
Yeah, I think the way to do it and how clinical labs are doing it, you have to have a certain rate of validation in these regions, and that's what you have to maintain no matter what your pipeline is exactly. But like as soon as you tweak a certain thing or whatever, you have to validate it. We have much more this discussion across pan-genomes, and if there is a way of integrating pan-genome into the analysis of clinical samples, and it's a nightmare, honestly, because like the tools are not ready yet, the whole framework is not ready yet. So like just to explain, also the pangenomic guys have currently like 3 or 4 different implementations of their pangenome where they cannot agree on their own of like which implementation is the right one. And so how should we ever bring this to a clinical if we don't know which one version we should use and how stable this version is besides not having tools for it. So I think it's going to be a big problem for sure. And if I know one thing, the clinical community is very conservative. I mean, like we are still using the same reference genome that we published like, 20 years ago, and not the one that was published 9 years ago. So there you go.
Anyone else have thoughts on the reproducibility? Here, I can bring the question back just so you can see. The reproducibility and integrating ML and how this kind of has this trade-off between interpretability and like good predictions, say. I think that's like related to one of our other questions.
For me, I think about reproducibility, I guess, like earlier in the analysis pipeline. A lot of what I do is at the discovery stage where I have a set of samples that are neurotypical controls and a set of samples that are Alzheimer's donors, and I'm trying to discover what the differences are. And here, the data are often put into repositories that are governed, like you have to apply for an account to be able to access these data, so it makes it a little bit easier in that sense. But I will say I have seen an enormous increase, happily so, with machine learning and AI even at this stage of the analysis pipeline, more at the discovery stage. And there have been some great products and like papers published thinking about reproducibility for machine learning in the life sciences. So I thought about a little bit of this. I published a set of guidelines or a set of reproducibility standards for anybody who is building a machine learning model or a deep learning model in the life sciences. And we thought about it as reproducibility is not black and white, it's a gradient. So what are some standards? So we thought about it as like a bronze, silver, and gold set of standards. There are, for example, places where let's say you build a deep learning model, how can you make others have this deep learning model accessible to others? So there's standard repositories, both outside of life sciences and in the life sciences for you to be able to contribute. But even so, I mean, there was like a famous paper from Google where, I'm forgetting all the details now, where they were showcasing their ability to identify breast cancer subtypes from images with a set of machine learning models that they built. And if you read their like data availability and code availability section, it's like, we use NumPy version blank, blank, blank. We use scikit-learn blank, blank, blank. And that was it. It was like the data are like everything. But at the same time, I also wonder like how much of it do we want that information out there in the public? And so, I don't know, I think it's a very tricky question. So even at the discovery stage, there's a lot of pushback on making your code and your data available to enable reproducibility, but I think there's been a really big push in the field to give tools for how to do that in a safe way that's like protective of the patient. So.
And I know, to add on to that, I think that the NIH is pushing now, similar to having to release all of your data on your genomes and all of the different pieces in a certain amount of time, they're pushing to have to release the code that you write for NIH-funded projects, because technically that is funded by the government, and so you don't own it. And so that's a big piece that I think they're gonna have to step in and do. My opinion on this is that, and I'm gonna plug Todd's vision on this, is that these things should be open source. We should, even with an ML model, you may not understand exactly what it did in the middle, but you can understand the code that went into it and how that was specifically written. And I think that the tools need to be available and open. I don't use things that are black box for a reason. I can't understand what happened to be able to disentangle what we just went through in my talk. And so I think that in a lot of ways, we publish the code that goes into our analyses. We publish all of that as a part of supplementals for our papers. And I don't think there's anything super proprietary in my code. The only reason that we have one project that we don't do that is because we're storing data in the cloud. And if someone knows the code, then theoretically they would have a better way of hacking into the system if they felt to. So that's more about controlling security than not releasing our data. The parts of the code that do the analysis are public. And so I think that, I think we do need to push for more transparency and reproducibility. And if I was a reviewer for that Google paper and all they said was scikit-learn and NumPy, going to go back and have them fill that out a little bit better, because that's not reproducible. You can't do anything with that. So I think that it needs to be across the board, but we need to be better about transparency of what we're doing, even if the ML model itself might be black box.
I think actually, this is complicated when we're dealing with clinical things, right? So this kind of leads to, again, one of the other questions, like when we are thinking about like kind of when there's more patient privacy questions, and I think even when you're mentioning, security, like code, well, of course, if people have to release their ML models, then we know that people can reverse engineer them. There are like privacy issues, but then code can also actually do this. So I guess then kind of one of the kind of related questions is, after implementing more sequencing initiatives within the clinic, how can we ensure the ethical dissemination of this knowledge among researchers and the clinician? And actually, since I don't know if Sonia, we could start with you, since I think we skipped you just now. The second question, after implementing more sequencing initiatives within the clinic, how can we ensure the ethical dissemination of this knowledge among researchers and the clinician?
Yeah, so regarding to the previous one, I totally agree with the data sharing is the solution for that. If you don't share the data, it's kind of high, and then make malinterpretations and things like that. I think that now the NIH, it's not score driving, but it's required, and they're going to get a more deep control on that, especially when you get funding. So totally agree. And with the ethic considerations, that is kind of challenging, because you don't know exactly, which points you can touch or not. And then, I don't know if that when we run the RV protocols, and then, when we write the consents for the patients and everything is well defined, but sometimes it's quite limited for resharing. We have blocks in some of the points because we cannot use some of the data, or we cannot get a whole interpretation or a single individual, because there are these limitations to get all the clinical data there. Like regarding, we have a limitation very strict in what kind of the data we can use or not. So it's a limitation for us in some way, but also, as you say, is protect the patients in the privacy in everything. So it's a line very well defined. Sometimes they don't care, but it's not writing on the consent, so they tell you things that they should not tell you should not know. But I don't know. I think that is depends on the way that we show the data and the interpretation that we present when we show the correlation between the demographic clinical settings and all the parameters with the sequencing analysis. And we have to be careful with that, because that can be quite serious as well. And making that, as much professionally and legally we can do it. But
Yeah, and I think I agree. And I think that, to clarify my last point too, I think the code needs to be available of how you analyze the data, but the actual genome sequencing data needs to be guarded as much as we possibly can. And I think, to add into this idea, how many of you are familiar with HeLa cell line and what that is? I think a lot of you probably are. So Henrietta Lacks was a woman who died of cervical cancer. And, yep. And one of the physicians took some of the cells from her to do some studies because it was considered medical waste, and they ended up being immortalized because of that cervical cancer embedded into the genome. And so they have been sequenced more than anything else. They've been grown more than any other cell line on Earth. And for a long time, this was publicly available information, but they found out that because this was a somatic germline kind of mutation, that this was heritable. And so her family now oversees the board to be able to get access to the genetic data. They are part of the process of approving what you can and can't do with that data, because it speaks to more than just the person whose data is there, right? And so I think we need to be able to be transparent about how we're analyzing the data, what the methods are, but then protect that data incredibly critically. And I think when we're talking about disseminating data and making sure this is ethical, we deal with things at the aggregate level, so it's not identifiable at the single patient level. This is stuff we do in epi constantly. And so I think the rules are kind of there for even genetic testing. We've been doing this for a while, but I think part of this is going to be technology is outpacing kind of the laws around this in a lot of ways. And so we probably need to circle back and strengthen GINA again and make sure that this is something that we have very strong ways of protecting patient privacy and rights, because, I mean, all of the examples of finding killers because of publicly available, lineage ancestry databases, like those things are possible, and more malicious things are possible too. So we need to be very thoughtful about this.
For the sake of argument. Come on, let's have a little bit fun. Obviously, I agree with you, but I mean, you have consortia now out there that have collected vast amount of genomics data. Again, I'm still on the human side. Which, even though if you're funded, it still takes you, like 8 to 12 months to get actually access to the data that you are funded to process, because of all the hoops, and like obviously the hoops are there for a certain reason, right? But we always have this kind of discussion, which is kind of interesting, between like, well, you're absolutely right. I can do really, really bad things with all these genomes that I have access to. What people often don't even discuss is like, I can actually predict the risks of your genetics risks down to the next 3 generations, up to the last 3 generations, and like cross-country through your cousins, no problem, right? Which is kind of a really nightmare, because like then your own consent form wouldn't be sufficient, but like you would need consent, all your relatives and all your families that you are aware of at least, which is kind of a nightmare, right? But like, I think on the other side, which was a really interesting question that I got once posed, is like, what do you do with a single genome that you have access to? What is the worst that you can do in reality? And that was actually really funny, because I was just applying for the green card, and I came back from my interview for the green card, and one of my center directors sat down with me. So Fritz, what would you do if you have the genome sequencing of the United States president? And I was like, is this part of my green card interview? But in reality, it's like, what do you do with one genome? Are you going out and say like, well, this guy has like 20% chance of getting a heart attack before he dies? Like, I'm just for the sake of argument.
Yeah.
Like,
I mean, so I'll, for the sake of argument with you, Alzheimer's risk. So if you have an elderly president who has a high risk for Alzheimer's, they're going to question every decision, and it can mute a lot of the decision making of someone who has a lot of power, right? So I think, you can do that with a single genome.
right, but like, I mean, like, you can argue that's fair, right? Like, if
Sure.
there are signs for neurodementia, then like maybe you want to do that. But I don't know the answer to it. Like, that's the fun thing about this question, is like, do you design a certain virus that goes after that person, or like, I don't know. I mean, like, no, go ahead.
Well, along the same line,
Yeah.
one thing that can happen is, say someone has the gene for Alzheimer's.
Right.
Like, maybe one of their family members can actually take away their rights prematurely, saying
Right.
that they may not have access, they may not be able to
Yeah.
make those decisions properly. So that can reduce life.
Right.
Life.
Yeah, totally. I mean, But our horror scenario is always like the health insurance hikes up your rate, right? This is always the U.S. health insurance nightmare. But like for me it's an interesting question, you know, because like I deal a lot with all these consent problems, and like there are huge problems with consents, and like some consent forms that were good like 2 years ago are not good anymore, and like we always have to review this part, and then what happens if the consent form isn't good anymore, but this sample is part of a collection, and like there's like such a depth, right? But like on the other side, on the other extreme is like really this question, like what do you do with a single genome if you want to do something bad to that person, or like some malicious stuff, which I find is also a very interesting question to pose. Of course, very counterproductive, but still interesting. Yeah.
I haven't watched it, but I heard that this is the premise for one of the recent James Bond movies.
There you go.
That there's some like genome-specific targeting assassinations or something like this.
They stole my idea.
Yeah.
I didn't mention it in the Green Card interview though.
Okay. Yeah. So if nobody else has thoughts on this, we can move on to our next question. How do you see the genomics field changing in the next couple of years? It's more.
I'm going to make this a little bit more fun.
Oh, there's a.
For the sake of argument.
No, it's a nice question, but I guess phrased differently, what is the one change or advance, if it were to happen, would either allow you to have like a breakthrough discovery in your group or retire and close your lab? So either or.
You want us to be put out of business?
Yeah.
No, I don't. I do not.
Is that
But
what?
I'm just curious, because I think that could give an interesting perspective on what really is holding back some of your research.
You want to take it?
I need a minute.
So I can shoot. I think what's coming on the horizon, and I think what's going to be reality in the next 5 years, is protein sequencing. Like, F all the rest with the genome and stuff, this is boring. Let's go to the proteins and really go in there and see what kind of proteins we have and what concentrations of proteins. And that would be much more direct with different phenotypes, right? The problem, though, is that this will change a lot between different tissues and also between different areas of tissues, right? So, like, what is your protein measurement good in your blood when you have actually neurodegenerative disease to be determined, right? So, like, this is kind of the thought process that I like to have these days. Is like, well, the genome is very indirect, right? And it's very comprehensive because everything is more or less in the genome, but it's very indirect. We actually look for mutations that manifests in mRNA, manifests in ribosomal transcription, and then manifests in protein folds or protein products and protein folds, and then there is some regulation, like 3 times along the way, but we're still trying to predict this. And then, like, well, there are other environmental factors of the cell and whatever not, right? So protein would be like a direct shot on your phenotype, more or less, right? But then how do you deal with the tissue specificity or the region of your, you probably know much more than I do about that stuff, but like, how do you deal with that stuff, right? You probably also have to deal with that when people ask you like blood samples versus this specific, like the left part of the hippocampus on Tuesday versus Wednesday or something like that. So like, yeah, that's what I'm thinking a lot about, like this promises of protein sequences and their challenges. There you go.
That's a good point. Let's see. So once I was in a training institute at one of the NIH institutes, I'll leave it anonymously, and one of the directors got up there and said, well, eventually we're just going to be able to measure all the data directly. Like, we won't need any tools to analyze anything because we'll just be able to directly see it and just immediately know what to do. And I was kind of like, what? That doesn't make any sense. There's a lot of noise in genomics data. So I think there's a lot of opportunities for new technologies, protein sequencing being one of them. I could go on about the technology itself, but the thing that I think about all the time is like, how can we teach at scale how to analyze these data in a way that is meaningful and a way that extracts information from the data. So genomics data is noisy. It is very, very noisy. And I spend my days training students and postdocs how to analyze the data. Some of my biggest breakthroughs have just been sitting in a lab meeting where somebody is presenting a new data set that they've generated and then discovering a better way to how to analyze these data. So for me, I don't have any sense of how to make that happen in the next 5 years. I just know new technologies will come, and it's really exciting, and it's a breathtaking pace to try and keep up with, I'll be honest. But it's very exciting, and I am very focused on how can we train people to be able to analyze these data and make sense of it. That's something that I'm very passionate about.
Yeah, so it sounds like, once you have protein sequencing, you'll still need to have single cell protein sequencing, and then single cell protein sequencing atlas, and then
We are like lawyers, like.
Yeah. Anybody else? What's the breakthrough that will close your lab, or that will change the face of the field?
Very positive vibe.
I've gotten this question is why I asked it.
Oh, you got this.
Google's going to put you out of business in 5 years.
Oh.
Maybe only for a couple of years. We'll see.
So I think mine would be, the thing that would let us make breakthroughs would be if software was supported for more than 3 years when it's developed. I'm so tired of reinventing the wheel. I do not want to reinvent the wheel. The wheel is fine. But the number of tools that we have used that die, like the switch over from Python 2 to Python 3, was brutal because nobody updated their stuff. And so I think that would be, the thing that would make life so much easier and let us focus more in on the biology, because we spend so much of the time focusing on that stuff.
If you're not aware, there's a funding agency, the Chan Zuckerberg Initiative, and they have a funding call the Essential Open Source Software, EOSS, I think is what it's called, and it's designed to basically support software that's widely used in the community and make it better. So if you have a favorite tool, ask your tool developer to submit an application so they can have funding to support that.
Isn't it the last cycle of that program
I couldn't tell you. I don't know.
I think it was funded for 2 years or 2.5
Yeah, but
or
that's one
something.
of the few. I mean, name
Yeah, no,
another
it
one.
is, it
Name
is, it
another
is.
one. I mean,
I actually found a lab equivalent to that. So we are having this Mendelian cohort stuff, and so we are trying to reanalyze unsolved cases to kind of identify causative alleles and report back to the families. And so part of the samples that were selected turns out are in the fridge since 15 years. And no one knows how they were prepped or when they were prepped. The PhD student is likely already a professor somewhere in the world. So yeah, sad story maybe, but yeah, also happens there.
Yes, I think these answers highlight how far the gap is from field-breaking breakthroughs. It's can we have the software be supported on Python 3? Okay, so actually, kind of a next question. What challenges will we encounter next with the vast amounts of genomics data already available or soon to be released?
So I'll start. We have more data than we can analyze at this point in time, and the scalability is just getting to be more. If you look at the amount of COVID data that is out there, there are millions and millions and millions of sequencing runs, some of them junk. Even just differentiating the good from the bad is hard, but we're in a point where, I was in a meeting at my postdoc institute, where it's a Human Genome Sequencing Institute, and they were generating more data than they could pay to store, and so the decision was made, once you analyze the data, delete it, because it's cheaper to regenerate it than it is to store it. And I was like, that seems terrifying to me. But we're in a position where we don't have scalable solutions for data storage, data analysis, the methods to be able to analyze these large data sets, to be able to leverage everything that's being put into NCBI. Whether or not NCBI is the right solution is a different question, but I think that's probably the biggest solution, or biggest challenge we're going to run into is the big data is becoming too big for current methodologies in a lot of ways. We're outpacing hard drive increases, everything Moore's law at this point in time.
Come on, single cell, you have to have an opinion about that.
You're right that data are big and they are becoming bigger. For me, again, it's not so much about the size of the data, it's about how can we develop tools to extract meaning from the data. I like to think about the most important about data science is the science, and how can we, again, extract meaning from that? So that's something that keeps me up at night all the time. I think big data is kind of a relative word in my mind. You are absolutely right, though, like that it's kind of astronomically large in certain fields, literally. And it's a big challenge. One of the things that I think I try to do, especially when I give talks like this, is try to bridge worlds. So in my own world, I am formally trained as a statistician at Rice. That's where I did my PhD. But I was also co-mentored by Sharon Plon at Baylor College of Medicine. And so I was instilled from a very early age, or very early stage of my career, the importance of communication and talking to the people who are generating the data. So for me, I try to pitch very hard, like the importance of learning two fields. And in that way, you can actually solve problems. While a lot of my colleagues, for example, in just the stats field, feel uncomfortable doing that, right? But I feel like this is how we're actually going to be able to take these large data and do something meaningful with it. It's not easy. It takes a really large amount of time and effort, and it's uncomfortable. But it's how I've been able to actually feel like I'm making progress in the field.
Okay, so I think we're almost coming to lunchtime. So I think one last question that was asked by the audience. As new sequencing methods are introduced and data sets shift with time, how do you think ML models trained with older data will be affected? So this is kind of relevant to this question of, do we just start throwing out the older data? Like, what do we, like, if we can't store it? But then how will that affect our models and predictions?
Is the question throw out, what
Well, no, they're not throwing that. How did the ML models trained with older data, how is it affected when there's more and more new sequencing methods, new data sets, new technology, I think, that are kind of appearing?
No.
I think it's interesting to think that I do think technology shifts do kind of spur on kind of new methodological changes. I mean, I think this, like in older examples, of course, the kind of migration from, say, microarray technology to sequencing technology, right? So it's like, how does that affected kind of our models, our kind of then moving forward?
Certain technologies give you a totally different resolution. I mean, like in my own field of gene expression, for example, microarrays are, you take a piece of tissue from my skin, it has millions of cells in it. If you process that on a microarray, what you get is one readout of all the genes from that across all the cells in that tissue. It's an average of expression across all the cells in that tissue. And it's just one readout. And then you could have a microarray run from your skin, my skin, your skin, and your skin. So we'd have 4 observations, for example, for all the genes that are expressed in our skin. Single cell was a totally different resolution. Now you get a readout of every cell. Now you get to see up to the full transcriptome of every cell. And so it's just, I think of it as like, well, you probably need a different model totally depending on what the resolution is or what the question is. I don't ever think we're going to have a machine learning model that's like one fits all for all resolutions. Maybe, I could be wrong. I would be super happy to be wrong, but I doubt that.
It would be really far down the analytical road. Yeah. Also, so for example, long reads, looping back, of course, to my own story, a little bit biased here. But there you have then also now epigenetics, which you don't get with short reads, which also like amplifies the whole thing. You can phase out single molecules in theory. And then like maybe also to the point of the previous discussion, like a lone single cell versus whole genome data, it's very different analysis because like Stephanie said, like single cell is much, much noisier. You have much, much more different amplification biases along the way or due to low coverage, 0.5x, 0.2x, a lot of the whole genomes at least, right? Versus like a 30, 40x coverage genome, like it's very different universes, I would say. Although it targets the same, well, maybe questions, but targets the same tissue at least. Yeah, so it's very, very tricky.
And I think when we're talking about clinical applications, when you see clinical tests come out, they freeze the test, right? They don't continue to iterate on it because it's been validated for a specific use case. So I think, when a new test comes out and is proven to be superior, gold standard testing, clinical trial, all that, then it replaces that, and you would have to have a new model for that. I mean, there's one idea is model drift. For bacteria, we see bacteria evolve microevolution levels across years and months and things change in the diversity of the organisms, new resistance mechanisms come out, so over time your model may be less accurate due to changes in the organism. It's not as impactful, I think, on the host side, but there's that idea of model drift, which is just that as the model ages, it doesn't reflect what it was originally, and then there's this idea of shifting to a fully fundamental new technology that will, because of differences in resolution, differences in the underlying assumptions of the test, require a new model. And so I think, to some extent that's progress, but we need to be thoughtful about applying these things and freezing them and having them be validated. Everything should be validated against actual biological things, not just things like recall, where it's a computational feature. It's important to know, but that doesn't necessarily tell you about the actual application, the accuracy, and say a test set.
I keep
Okay.
thinking if I could throw at you animal models with that question.
Ah. Well,
Yeah,
I think
probably
in
not.
general, though, it seems like with this question also highlights that we probably won't be put out of business soon. It's like, there's new technologies, there's new data, there's even biological drift. So I don't think we're anytime soon getting the one model to rule them all.
Yeah, it's I've figured it out, but why is it that way, right? There's always that next question, so.
Yeah. Anyways, okay, so we should kind of wrap up and go to lunch. Thank all of our panelists. I don't know, any final words if you guys have? Or you can chat with them at lunch. Or Todd, if you have any.
Yeah, I mean, again, this I think was fascinating. One thing I just thought of as hearing our panelists was one thing related to what Fritz said. 200 copies represents a disease state. Why is it 200? I guess you didn't address that. I mean, back like interpreting 200 copies of with Fragile X was 200 and not
Oh,
201
that
and
stuff.
199. We
Yeah.
have a very strict threshold.
We have these thresholds, yeah.
In the future, for new stuff, how do we learn what that copy number is? Because you said there's a range, right?
Well,
When we
that,
look-
yeah,
Anyway, we can talk about it over lunch. My comment is.
is
putting
a
all
distribution,
together,
yeah,
I
yeah.
think there's idea of what we heard in what Stephanie said for the Atlas type analysis where we just establish baseline of what we know, and then we go look for, do these things occur? But then there's an idea going back to what Blake said about making it affordable. Like, I think there is a role for that, right? We just can't sequence, as much as I always put this in my grant application, sequence everything. At some point it has to go to like affordable sensing technology, or or like physicists do, kind of just toss out 99.99% of the data and look for one.
But like honestly, how is it that like the medical system charges like, I have no idea what, $20,000 for an MR or whatever, but we have to deliver a test that costs 50 cents. Like there's a little bit of a gap between that, right?
My comment is more on the Atlas building side, right? How do we build that? Because I've not found a single grant application that would give me enough money to do any of this, even for a small cohort. We have a population of 2,000 patients, and we're spending all of the money, and we don't have enough that we were given to actually do the sequencing for that. So there is a disconnect between, I think, the money required just to do that. But on the test side, that's a whole different equation. I don't know what the test is. I've seen tests that if they're not less than $100, they're not feasible, and they're using, other equivalent tests cost thousands of dollars. I don't understand how to square them. But anyway, my comment just is trying to bridge all of the feedback, was it sounds like we're keeping our jobs. There's a lot of really open, important questions, but there is kind of a lot of really important ethical issues and considerations to keep in mind. Just coming back to what Sonia said about really even like trust and understanding, IRBs and trying to, how do we make this as lean as possible while still facilitating research and application. Anyway, thanks again, great discussion, and now I guess we look forward to lunch.
Yeah, I think I think we had somebody had one question, audience question. Feel free to accost, I think, the panelists to ask them about streaming technologies.